David Martinez Selva. I'm a senior scientist working at the Diabetes and Metabolism Department at the Vall Research Institute in Barcelona, Spain. And I'm going to talk today about the sex hormone binding globulin and its regulation by nutritional factors and the role of this protein in obesity development and progression. First of all, I don't have any conflict of interest. And for the ones that you don't know, this protein called sex hormone binding globulin, it's a 90 kilodalton homodimeric glycoprotein produced and secreted by the human liver. And it's secreted into the blood where it binds with high affinity androgens and estrogens. And it regulates their availability at the tissue and cellular level. Low plasma SHBG levels are a biomarker of different metabolic diseases. And in fact, you can find low plasma SHBG levels in subjects suffering inflammatory diseases, hypothyroidism, obesity, and polycystic ovarian syndrome. More importantly, epidemiological studies have demonstrated that having low plasma SHBG levels, it predicts you a higher risk of suffering both cardiovascular disease and type 2 diabetes. On the other hand, you can find subjects with high SHBG plasma levels suffering from anorexia or hyperthyroidism. So in my lab, we're interested in knowing which factors regulates SHBG and induce this reduction in plasma SHBG levels as body mass index increases. And also we are interested in knowing if the reduction of the plasma SHBG levels in these subjects is affecting also to the progression of the disease. So we use the most important animal model, that is the mouse, and for different reasons. But it's also important to know that some genes are not exactly, they don't have the same gene structure as in humans. And in fact, the human SHBG gene is one of these genes. This is the structure of the human SHBG gene, which it has a regular promoter which drives the expression in the liver to produce the SHBG, but also there is an alternative promoter with alternative exons that drives the expression of the gene in the testes, especially in the germ cells. If you check the mouse gene, it has differences at the promoter region, and it also have the 8 exons and introns. But this gene in mice is expressed in the Sertoli cells of the testes, which means that the mice do not have SHBG circulating in the blood, and you cannot study in rodents or lower mammals the expression of this human gene. So what we did several years ago is to create a human SHBG transgenic mice. But in the lab, we use different models and samples. So we used a HepG2 cell line that is a human liver cell line that expresses and secretes into the media the SHBG. So we can monitor, if we treat these cells with different factors, we can monitor the accumulation of the protein in the media. We also use the humanized SHBG transgenic mice. These mice express the human gene and they secrete it into the blood. So we can treat these mice either intraperitoneally or with different factors in the drinking water, and then we can follow the plasma levels in the blood of these mice. And finally, we have access, since we're in a campus in a hospital, we have access to human biopsies from liver and adipose tissue to corroborate our findings in vitro and with our model. So for the last 10 years or so, we have used these models to determine, for instance, how high carbohydrate diets reduce the plasma SHBG levels through an increase in hepatic lipogenesis. We also have shown that different factors of the Mediterranean diet, such as olive oil or red wine, can increase the plasma SHBG levels. And also we have identified different factors produced by the adipose tissue, such as pro-inflammatory cytokines, how they reduce the plasma SHBG levels, and also anti-inflammatory cytokines such as adiponectin that increase the plasma SHBG levels. So for the rest of the talk, I just want to point out that it's going to be important to understand the results, that this promoter sequence that drives the expression of the gene in the liver, have different footprint regions where different transcription factors can bind. I'm not gonna talk too much about it, but just you have to remember that PPR gamma represses the expression of the SHBG gene, and HNF4 increases or activates the expression of the SHBG gene. So several years ago, we wanted to study how high carbohydrate diets affects the SHBG production, and the first that we did is use our in vitro model, and we give these cells different amounts of glucose or fructose, and we saw that the accumulation of the protein was reduced when you treat it with carbohydrates. We checked that at the RNA level, and then we saw that the high carbohydrate diet reduces the amount of the HNF4, that it's the activator of the transcription. Then we repeated the experiments with a different amount of glucose and fructose, and we saw that this increase in carbohydrates, what did is increase the hepatic lipogenesis, and you can see here the amount of palmitate that these cells accumulate. And in fact, if you take these cells and you treat them directly with palmitate, you can see that you reduce the production of the SHBG protein. So we next went to our in vivo model, and we give our transgenic mice, either males or females. We give them a normal diet. The SHBG levels are more or less constant. And then when you switch these mice to a high sucrose diet, you can see that in 7 days, you have a reduction almost of 50% of the SHBG production. And when you switch them back to normal diet, the SHBG levels are recovered. And the same with the males. Then we repeated the experiments, but only for 7 days, and we give different diets: a high glucose diet, a high sucrose diet, and a high fructose diet. And as you can see, the high fructose diet is the one that is reducing more the production of the SHBG protein in the liver, and that's because the fructose is more lipogenic than sucrose or glucose. So why that is important? Well, if you check the amount of sugar that the Coke has, for instance, the amount of sugar that you have in a can of Coke, it's 10 teaspoons. And I don't have anything against Coca-Cola. So if you check the different energetic drinks, you can see that the amount of sugar that they contain, it's huge. And not only beverages and sodas, also orange juice or even the ketchup that has a huge amount of sugar. So our point was that if I'm not a nutritionist, and I'm not gonna talk too much about it, but if you have a normal menu that you can have your carbohydrates in the potatoes and green beans, you have a steak, and then you have orange and water for drink, that's a good menu. But if one day you decide, okay, instead of water, I'm going to drink Coke, and instead of having the meat alone, I'm going to put some ketchup, and today I'm going to do an extra, I'm going to eat an ice cream for dessert, you can see that this excess of sugar is what it's affecting the liver. It's increasing the lipogenesis, and it's reducing your plasma SHBG levels. So the excess of carbohydrates that we're eating with certain foods are really important. So next, what we decided to study was the different ingredients of the Mediterranean diet, and because it's known that Mediterranean diet reduces your risk of cardiovascular disease. And since we saw in the first slides, if you have low levels of SHBG, you have a higher risk of suffering cardiovascular disease. So what we want to study is if there are different components of the Mediterranean diet that increases your plasma SHBG levels. And we started with the olive oil. So several years ago, another researcher that we collaborate with make a study in a small village in the south of Spain, and they give them during 6 months, the whole village, they give them olive oil to cook and to eat. And then they check the SHBG levels before and after the 6 months, and we saw a clear correlation. As the olive oil consumption increases, you have higher levels of SHBG. Then we went back to the lab, and we give to our in vitro model, we give them linoleic acid or oleic acid, and we see that oleic acid increases the plasma SHBG production from the liver, also the protein and mRNA levels. And when we check the molecular mechanism, we saw that the olive oil consumption, what it does, it decreases the PPR gamma protein levels and RNA and protein levels. So you have that's why you have an increase in SHBG production. After the olive oil, we check the red wine, that it's another component of the Mediterranean diet. And here what we did is we diluted white wine and red wine, and we give it to the cells for 3 days. And to our surprise, we saw that red wine increases protein levels almost 3 times. And also this was confirmed at the RNA level. So we start reading about, because I don't drink, so I didn't know anything about wine. But we start reading, and we saw that the difference between red wine and white wine sometimes is the amount of resveratrol. So we took this resveratrol and we give it to the cells, and as you can see, we can mimic the effect of the red wine at protein and mRNA levels. So we next wanted to check the transcription factors that regulate SHBG production in the liver, and when we check HNF4 or PPR gamma, they weren't changed. So we went back to the literature, and nevertheless we give the resveratrol to our mice in the drinking water, and we saw that the resveratrol didn't have any effect on the SHBG levels when compared with the controls, and also the mRNA levels weren't unchanged. So we went back to the literature and checked which genes in the liver are responding to resveratrol, and we checked 2 different genes, the CPT21B10 and the UGT1A1, and we saw that in our mice, the mRNA levels of these genes were increased. So the resveratrol was having an effect, but it didn't affect the SHBG gene. So we went back to the literature again and started reading about resveratrol in mice, and we saw that resveratrol can bind one receptor that's called the constitutive androsten receptor, and this receptor have different affinity for different compounds in mice and humans. So we transfected our cells with the human CAR and with the mouse CAR and repeated the experiments. I'm going to go in too much detail because we use an agonist and an antagonist, and we demonstrated that the effect of the resveratrol in the HepG2 cells was made through this CAR receptor. So next, what we did, we bought a humanized CAR mice, and we crossed it with our humanized SHBG transgenic mice. So we have mice that express SHBG, human SHBG with a humanized CAR, and then when we give the resveratrol, we can see the increase in SHBG plasma levels. So now, next, I'm going to talk about if an individual starts gaining weight and develop obesity as its SHBG levels go down, if this reduction in SHBG is affecting the progression of the obesity, and how we study this. So first of all, we make a new mouse model that by crossing our humanized SHBG transgenic mice with the obesity and type 2 diabetes mouse model, that is the DBDB mice that have a point mutation in the leptin receptor, and when you have this mutation in homozygosis, these mice are hyperphagic and they eat all day, and they develop obesity and type 2 diabetes by 4 weeks of age. So when we characterize these mice, what we saw is that the obese and diabetic mice that have these mutations that develops obesity, when you introduce the overexpression of SHBG, you can see a reduction in about 15–20% of the weight of these mice. And as I said, by week 6 they develop diabetes, and the overexpression of SHBG does not affect to the glucose levels. So next, what we saw with these mice is that they have less less amount of adipose tissue, both in males and females. So in order to corroborate these studies, what we did another experiment where we study the obesity development using a high-fat diet during 8 weeks. So we give Walter mice and SHBG transgenic mice a high-fat diet, and as a result of these studies, what we saw is that humanized mice that have SHBG in their bloods, when they eat high-fat diet, they are protected against obesity development, and they increase the same amount of weight that wild-type and SHBG mice eating a controlled diet. So in order to corroborate that they are eating the same as the wild-type mice, we put these mice in metabolic cages, and as you can see, the daily food intake and the water food intake. Water intake was the same, and the production of feces and urine was also the same, and we also checked the lipid absorption at the intestine level, and they absorb the same amount of fat that the wild-type mice. So how SHBG overexpression prevented the increase of obesity? We also checked that SHBG overexpression protected against the hyperinsulinemia induced by the high-fat diet, and also protected against the hyperleptinemia induced by the high-fat diet feeding, and the hyperresistinemia. Also, we saw that the SHBG overexpression increases the adiponectin production by the adipose tissue. So then we also checked the amount of adipose tissue of these mice, and we saw that the adipocytes were smaller, and how SHBG make this reduction in adipose tissue. So we found that SHBG induces lipolysis in the adipose tissue as detected by increasing phosphorylation of pKa and phospho-HSL. Also, when we went to check the brown adipose tissue, we saw that the overexpression of SHBG protected against the differentiation that suffers this tissue when you are eating a high-fat diet. And also we can saw that increases the levels of UCP1. So the activity of the brown adipose tissue in the mice that overexpress SHBG was clearly increased. Okay, so that's everything. That's a resume of all my talk. And if you have any questions, I'd be happy to answer it.